Let's dive right into the blueprint of life. In today's explainer, we're basically going to map out a 3D animated journey through the cell. We're going to visualize every single critical event during the cell cycle, mitosis, and meiosis, breaking down the entire process so you can absolutely master this topic for your exams.
I want you to just picture zooming into the microscopic world of one of your own blood vessels. Literally right now, every single second, millions of your cells are actively dividing. Some are working round the clock to replace damaged tissues, while others are tasked with the incredible job of creating entirely new life. But how exactly does one single cell become two, or even four? Well, that's exactly the journey we're going on today.
Here's our roadmap for the explainer. We'll start with the overall cell cycle journey. Next, we'll look at interphase, where the cell preps to divide. Third, we hit mitosis, which is all about growth and repair. Fourth is meiosis, the creation of unique life. And finally, we'll wrap up with a head-to-head comparison of the two.
Okay, section 1. Let's start our arc at the very beginning of a cell's daily life. So, at its most basic level, a cell is basically a membrane, some jelly-like cytoplasm, and a nucleus that acts like a vault, holding our DNA. The cell cycle is just the sequence of events where the cell grows, copies its genetic material, and then ultimately splits. Broadly speaking, it's divided into two major chunks, interphase and the mitotic phase. Think of a cell's timeline kind of like a continuous clock. It's just rotating constantly through four specific slices, G1, S, G2, and the M phase. The cell moves through these stages in a highly regulated non-stop loop.
Let's move to section 2 and see how this actually builds. We're entering interphase, which is absolutely the longest stage of the cycle. This is where our biological factory gathers its resources.
Imagine the cell physically plumping up and enlarging as its internal organs, the organelles, start to multiply. Mitochondria are increasing in number, proteins are being pumped out, and this all happens across three key steps. Step 1 is the G1 phase. The cell is just growing, doing its normal day-to-day job. Step 2 is the S phase, where DNA replication happens. And step 3 is G2. Think of G2 as the ultimate quality control inspector. The cell actively checks for DNA errors, fixes any damage, and duplicates its centrioles to get totally ready for mitosis.
Now, the most crucial moment in that S phase happens when the double helix literally unzips. As replication kicks off, the cell rapidly creates two perfect, identical copies of every single chromosome. Once this happens, there's no going back. It is the ultimate point of no return for cell division.
So, having fully prepared, let's jump to section 3 and explore mitosis, growth, and repair.
Before we get bogged down in the dense, phase-by-phase terminology, we really need to anchor our understanding on the specific purpose of this exact type of division. Mitosis is all about your body's growth and tissue repair. The whole goal here is to create genetically identical daughter cells. Zero variation, just perfect clones designed to heal a cut on your finger or grow a piece of bone.
To pull this off, the cell goes through 5 highly sequential steps: prophase, metaphase, anaphase, telophase, and cytokinesis. We're going to walk through each one right now. Think of it like a mini story of cellular action, almost like a camera panning smoothly through a microscopic battlefield.
First up, prophase. The loose, stringy chromatin condenses into thick, visible chromosomes, and that nuclear membrane completely breaks down. Next is metaphase. Think M for middle. Those chromosomes line up perfectly along the cell's equator, and these little spindle fibers attach to their centers. Then anaphase kicks into high gear. The fibers aggressively shorten, pulling the chromosome halves, the sister chromatids, apart toward opposite ends of the cell. Finally, in telophase, 2 brand new nuclei reform around those separated bundles.
But hey, the cell is still technically one giant structure until we hit cytokinesis. That's when the cell physically pinches inward, creating a furrow that gets deeper and deeper until, snap, it perfectly splits into 2 genetically identical daughter cells.
All right, moving to section 4, we are now leaving the realm of basic tissue repair and entering the wild world of meiosis, which is all about the creation of unique life. Keep in mind, this process introduces profound genetic diversity. Unlike mitosis, meiosis has one job and one job only: creating specialized reproductive cells, which we call gametes, like sperm and eggs. To pull this off, it actually requires 2 back-to-back cell divisions, which ultimately output 4 genetically unique cells.
Now here's the vital piece of genetic math you absolutely need to remember. A standard human body cell has 46 chromosomes, but a reproductive cell, it only has 23. Meiosis literally has to reduce the chromosome count by exactly half. Why? So that when two reproductive cells eventually combine, the resulting life gets right back to that correct magic number of 46.
To make this math work, meiosis happens in two massive waves. The real magic? That happens during Prophase I. Homologous chromosomes pair up and undergo something called crossing over. DNA segments dynamically swap maternal and paternal material, which is what guarantees you don't look exactly like your siblings. After that, the paired chromosomes align, separate, and form two haploid cells. Then a faster second wave hits, Meiosis II, which is a lot like standard mitosis, where the sister chromatids in those two cells are pulled apart.
4. Remember this number for your exams, because Meiosis II speeds through that second division. The final, beautiful output isn't 2 cells, it's exactly 4 totally unique haploid cells.
Okay, let's wrap this up in Section 5 by synthesizing all this complex biology and putting these two processes head to head.
Let's lock in these contrasts. Look at how they flow side by side. Mitosis takes 1 division, meiosis takes 2. Mitosis gives you 2 cells, meiosis gives you 4. Mitosis produces diploid cells with a full genetic set. Meiosis produces haploid cells with half. Mitosis builds identical clones, meiosis builds completely unique cells. And functionally, mitosis is just for growth and repair, while meiosis is strictly for reproduction.
Zooming way out from these microscopic dividing cells, we can really see the macro, real-world results of all this. We see human bodies miraculously healing from injuries thanks to mitosis. We see vast forests growing and animal kingdoms reproducing across the planet thanks to meiosis. The silent, rotating DNA inside us literally translates to all life on Earth.
At the end of the day, cell division is just nature's most remarkable process. It guarantees that life doesn't just heal, but it continues and adapts across generations. As trillions of your very own cells are flawlessly executing these beautiful microscopic cycles right this very second, it leaves us with one pretty inspiring thought. How will you use the life they build? I really hope this explainer helped you marvel at the breathtaking biology happening inside you right now. Keep learning, and I'll catch you in the next one.